Visualización, ¿cómo se dice? Compartir pantalla, María. Pantalla dividida, pantalla completa. Llamada. Compartir pantalla.
Pero usted no le puede compartir a él.
Él tiene que compartir.
Dale cancelar. Dígale que en call.
You need to go to call.
In the menu.
In the menu bar. And then you have to share your screen.
Yeah, so let me just get in there. I think I can't do it here. So I've got to transfer I've got another computer here that maybe works better. So I guess I'm having some problems.
Okay, no problem. Take your time. People is coming. So we have time.
If this works. So let me maybe try to, I'm going to hang up. And then I'm going to call you back in 1 minute to see if I can do this.
Aquí.
So I'll call you back in 1 second.
Okay. Va a cargar de computadora.
Ahorita se está transmitiendo.
Por el canal de YouTube.
Lo que hay que hacer es ver acá. Se le da detener transmisión cuando quiera detener.
Parémoslo ahorita.
Okay,
So,
we're back now. Does this one work okay? Can you hear me?
yes, I can hear you. I can see you.
Okay, great. So what I'm going to do is try to open my
You need to go to the menu bar and then call and then share screen.
Okay, I'm getting there. I get. So I need to go to share screen, and where is that?
That is over in the menu bar. You have to go to call. That is Skype, contact, chat. Up.
Microphone, add participant, share screens, okay. So, and let me get to the
Then you have to open your presentation. So we can.
Yeah, I'm getting there. Yeah, so let me just go back to. Oops. Go up one. Okay, so now.
Now I can see your presentation now.
Okay, great.
So
So this is the same one from Vienna, but I can talk in a slightly different way about coffee and other crops.
And maybe also rice, because we have another project with rice.
Sure, yeah,
No,
we can stress that too. Okay. Good, okay.
Ah, él lo tiene ahí. Él tiene. You have the window in the screen.
So
This window
Yes.
here,
Yes.
Uh-huh.
I have to put that, maybe that's okay.
Right there. It's okay. So I'm
So I
going.
can still see it, I can still have the microphone controller. Is that okay?
Yes, it is okay. So I'm going to present you in Spanish. It's okay.
No problem.
And then you can talk tal vez le podemos enseñar. Muy buenos días. Gracias por asistir a esta videoconferencia. La vamos a tener con el doctor Brad Hill de la División de Mejoramiento Genético de la Agencia Internacional de Energía Atómica. Ya tuve la oportunidad de compartir con él hace 15 días un curso, un entrenamiento en inducción de mutaciones, específicamente en café, y le pedí que si él podía compartir con nosotros las nuevas tecnologías que se están implementando para la detección de mutaciones. Vamos a tener una charla aproximadamente de 40 minutos y al final, si tenemos preguntas, las podemos hacer. La charla va a ser en inglés. Él solamente sabe algunas pocas palabras en español, pero no puedo. So Brad, I'm going to show you how many people there are here.
Great.
We are about 15-20 persons.
Great.
So if you want, you can start now with your
Okay,
presentation.
I can start. Well, buenos dias. I want to thank Andres for organizing this, and thank you all for coming so early to the lecture, because for me the sun is already setting and it's getting dark and cold here in Austria. What I wanted to describe to you are some of the tools we've been developing that are helping us use mutations to improve different crops. And I'm giving some slides that I gave at a presentation in October that Andres was at here in Vienna, and I'll add to this a little bit on different crops, and I can comment more on this. So it really focuses on next generation sequencing technologies and the use of induced mutations. So I hope you can see this slide. This is a pie chart where we are looking at the different crops that have been produced using different types of mutation. We have a database in Austria that we house, and you can visit that here on the URL. We have now registered in our database over 3,200 mutant crop varieties. Oops. The majority of them are produced with physical mutagens, the most common being gamma radiation. X-ray radiation is another tool that's often used, as well as chemical mutagenesis, and they do different things to the plant genome. Chemical mutagens, the most popular ones are EMS, and also sodium azide and MNU. If we look further at this pie chart just a little bit, we can see that rice represents the major crop that people have successfully used for mutation breeding. Rice is an important crop all over the world. It's the most important food crop and food staple to help avert problems with food security. So we have 25% of the rice of the population is rice, followed by barley, and then wheat. So these three major cereals represent the major food varieties that have been produced. But we can also see here chrysanthemum, which is an ornamental flower, soybean and maize, and then other plant species. There's examples of over 200 plant species that have been improved through the use of induced mutations. Just one example that I can give you comes from Peru, where the folks at La Molina in Peru have used induced mutations to improve both barley and amaranth. What they've been able to do is use gamma radiation to, for example, in barley, to get barley produce a yield increase of over 6-fold, and at 3,500 meters and above. So these are crops that normally produce very poorly at these high altitudes, but using induced mutations, they can improve them. This has resulted in over $6 million US being added to their annual local income. So we see a nice example of that, and because of this achievement, they were given an award from the FAO and the IAEA, which is the program I work for, in 2014 with Professor Luz Gomez Pando, and she's in the center here somewhere. Interestingly, we don't know why these plants perform better at high altitude. So we know they've been mutated. We don't know what happened in the genome, and that's some of the work that I've been focusing on and working on for the past several years. So if we just think about how people do mutation breeding, in this example, I'm using rice because seed-propagated crops really predominate in mutation breeding successes, and that's because they're relatively easy to use. So with traditional mutation breeding or forward genetics, we simply take the seed and we apply a dosage of irradiation. And this is optimized ahead of time. You basically put the seeds in a machine. It's almost like a microwave. You apply the energy and then you grow the plants up. Rice, as an example, is self-fertile. So the first generation, these plants are genotypically heterogeneous. There's many cells in the rice seed, and each cell will accumulate different mutations. That means that this plant, this first generation plant, which we call the M1, each leaf has different mutations in it, and not all the mutations are heritable. So this is not very good material to work with. We work with, starting with the M2 generation, where we expect Mendelian-type segregation of the mutations. One normally applies phenotyping and then tests that for many years to make sure the phenotype is stable. In many countries, you undergo a varietal release program that involves multi-location trials. After that, and through government certification, you can create a new variety. Later on, people may ask the question, what's the gene that's affected? And this is where we adapt molecular tools for. We can also do reverse genetics, and reverse genetics is basically the same idea, but in reverse. Instead of looking at the phenotype first, you look at the genotype first. So you make the same kind of mutant population. Normally, people are using chemical mutagens for reverse genetics because they produce point mutations in the genome. After you make your mutant population, you can apply some molecular tools to find mutations, ask a specific question, test a hypothesis about gene function, and ask what happens when you disrupt a gene function through mutation. What's the phenotypic consequence of that? Now, we use molecular tools to optimize the efficiency of these processes. First of all, we can develop methods to accurately test how many mutations we're inducing for a given treatment. And you could imagine if you have a dosage of gamma radiation where you produce twice as many mutations, you can have your success with half as many plants. So you can increase the efficiency of this pipeline simply by doing a better job of inducing mutations. And the same is true for reverse genetics. We are also developing tools that help us clone the genes that are causing the traits, and also finding the mutations in the genes to speed up reverse genetics. And I'll give you some examples of each of those in this presentation. So the first example comes with rice, and we have a project through the International Atomic Energy Agency with Madagascar. In Madagascar, they're interested in a number of traits, of course, yield and quality, disease resistance, and also parasite resistance to striga. So what we did here is we started a mutant population, and this is the visiting scientist named Lydia, who came from Madagascar on the right panel at the bottom. Joanne and my group started this work with Lydia, and they created mutant gamma and X-ray irradiated populations up to the 7th generation. So they took this through 7 cycles. They then did forward genetics to look at the phenotype. So this is a very traditional approach. And here's some photos of what this looks like. So these were planted at the same time. On the left of the first panel is the mutant, followed by the parent plant. And you can see the rice plant has a shorter stature. It also matures earlier. On the right-hand side, you have the mutant and the parent panicle. And what you probably cannot see too well is that the seeds have a darker brown color and they're smaller seeds. So we see fairly different changes in the adult plant We then decided to take a whole genome sequencing approach. So we used an Illumina platform to sequence all of the DNA in these plants, and then did some analysis with that. And we used an analysis called Bin by Sam, which was developed by our collaborators Luca Comai and Isabel Henry at the University of California, Davis in the United States. This is what this data looks like. So on the top, we have a panel showing a snapshot of part of a chromosome. And all of these lines here represent DNA sequence data. The number here is set to 2, and that's because rice is a diploid plant. So what we can see as we look from left to right here is we see the data and the DNA sequencing reads at 2 copies, except in this region where we see a drop, where 2 lines drop down to 0. This is representative of a homozygous deletion that was caused by the gamma radiation treatment. We mapped the size of that, and that we expect to be around 150,000 base pairs. So a rather large deletion covering almost 40 genes. And Bernard Hoffinger, the man in the photograph in the center here, then decided to create a rather simple approach to make a genetic marker for this so that we could use this in breeding. And this is the figure down here. So this is a traditional cheap agarose gel. And we have these bands here representing PCR product from the parent. And the lanes labeled N are the mutant lines, where we have this new variation where we no longer see an amplification project, indicative that the DNA is missing. And the expectation is that's caused by the treatment with gamma radiation. We're further characterizing. So we're here now with this kind of mutation work. We're refining this work a little bit. This work was done with relatively low-cost whole genome sequencing at low coverage. Joanna and my group, and just before this Skype conference, we were meeting to look at new data. So we've collected what's called high-seq data. And we are also doing metabolomics and proteomics with collaborators at the university here in Vienna to better characterize the mutant plants. So here we have an example of tools that we're able to develop in the lab to quickly analyze what happened when we've mutated a plant and to try to understand the genes and test the genes to decide if those mutations are really causing the phenotype. Now, one of the things that we've done also is, so with rice, the rice genome size, effectively what we're sequencing when you remove Central American telomeric sequences, we're sequencing around 400 million base pairs. That's down here. This is in blue is the size of plant genomes, and this is in hundreds of millions of base pairs. So Arabidopsis is about 135 million base pairs. You can see that the size of plant genomes, starting with about soybean and definitely with maize, gets very large. And the problem we have is it's very expensive to sequence the whole genome of something like wheat to get enough data to really analyze for mutations. So what we looked at is to try to develop methods where we sequence only the genic regions. And we used a method called exome capture sequencing so that we only look at the genes. The exome size is about 80 million base pairs. And it's constant, more or less, in all plants. So if I want to look at the gene space of wheat, I need to sequence around 80 million base pairs. If I want to look at that in barley, around 80 million base pairs. If I want to look at that in sorghum, it's about 80 million base pairs. So that's a size that you can do relatively cost effectively with minimum equipment. So what we decided to do is test 2 species. We looked at one seed propagated crop, which is sorghum, which we had a lot of experience with, and we used 2 different varieties from Sudan that we mutagenized in our own laboratory. We propagated this in our field, and we took 15 plants representing different treatment dosages. And then we also did gamma and EMS mutagenesis of cassava, which we vegetatively propagated. So we propagated this in tissue culture, and we took 17 of these selected for sequencing, And the work here was done by Bado in our group. Oh, and these guys are former members of the lab, and Joanna as well. So and then we decided to do this sequencing with exons only, and we see some very nice results. So again, we're looking at sequence reads from the genome that are adjusted for the dosage of a diploid. So this is sorghum. Sorghum is a diploid. And you can see this region where two plants that are siblings of each other, that are genetically related, are missing DNA. And it's presumably due to the mutation induction process. This is another example. This is from chromosome 1. We see both homozygous and heterozygous deletions. These are two examples of heterozygous deletions in material that was treated with 75 grays of gamma radiation. They were quite large deletions, about 575,000 and 400,000 base pairs. We did this, we looked at this, we were working with our colleagues in California to look at this data, and we see the range here in the third to the right column is the size of the different lesions we find. Anywhere from 100,000 up to nearly 700,000 base pairs in sorghum. We found both insertions and deletions. We found mutations in 4 of the 5 analyzed mutants in one sorghum line called ABU, but another line that we did at the same time, we found no mutations, which is really striking to us. It suggests that there's cases where we're unable to induce mutations efficiently in some genotypes, and we don't understand that yet, but what it does mean is we can fail at mutagenesis, and that we need to pay more attention to this when we're doing our experiments. The other thing that was striking to us when we think about tissue culture mutagenesis, and this is something that we are doing in cassava and in banana, and that we're thinking about doing in coffee, we really found no mutations. Both with EMS and gamma radiation, we tried a number of different technologies, and we really couldn't find anything that was believable. So we think there's something to do with the meristematic organization in diploid cassava that's somehow recalcitrant to accumulating mutations. Oops, forget that slide. I'm going to go into tilling now, and then I'll show you one of these examples. So we've done the same kind of work with banana, and that was a slide there that we have new data in banana showing deletions of up to 3.8 million base pairs in banana, which is kind of exciting. The third example, we'll talk about reverse genetics a little bit. The reverse genetics method known as tilling was created by Claire McCallum when she was a graduate student with Steve Henikoff in Seattle, Washington. It stands for Targeting Induced Local Lesions in Genomes. What she did back in the late 1990s was she made a mutant population of Arabidopsis thaliana, and she used the chemical mutagen EMS for this. And her logic was as follows. She thought that if you could make a big enough mutant population, a large enough population, and that each plant could harbor many mutations, you could make a library, and if you had enough plants, you could guarantee multiple mutations in any gene in the genome. So in effect, you could have a seed bank and a DNA bank where any question you wanted to ask about mutations in a gene, you could ask with a single population. This was followed, then the strategy then was to take plants to the M2 generation using a single seed descent strategy to maximize the mutation diversity, make the DNA, and then at that time use PCR-based methods and the mutation discovery methods to find the mutations. And we've been doing these kind of experiments for years now, starting in Arabidopsis and then expanding to many different species. There's now more than 23 species for which people have published tilling on. I provide only a little bit of data here from EMS to just show you one point, and that is what we do know is with pretty good prediction what kind of mutation densities we should get. Most of the mutations caused by EMS are GC to AT changes. And we know that in diploids, we can expect mutation densities from about 1 in 150,000 base pairs to about 1 in 500,000 base pairs. So this is the range for diploids. Polyploids are actually easier to mutagenize because they have multiple copies of the gene. So you can accumulate more mutations before you reach lethality. This is something actually that Louis Stadler first showed with wheat in the late 1920s. And we see this here with spling populations as well. This is data from Ann Slade's papers. Tetraploid and hexaploid wheat, you typically get mutation frequencies of about 1 in 40,000 base pairs and 1 in 20,000 base pairs. So we know that if we can estimate the mutation densities, we can effectively estimate the size of the population we need to be successful. Something like Arabidopsis, we know that a mutant population of between 3,000 and 6,000 plants is sufficient to find deleterious alleles in most genes in the gene. People have been using reverse genetics to improve traits that are agronomically important. And this table just shows a few examples. For example, high amylose starch in wheat caused by inducing mutations in SBE2A. Plant height can be affected, of course, with the hormone pathways, gibberellic acid, and this was shown in P. Content for industrial use by mutating granule-bound starch synthase was published. Fruit color in tomato. We have some new mutants in tomato that are heat tolerant as well, and then there's altered hydrogen cyanide potential in sorghum to improve its feed quality for animal feed. So one of the things that we wanted to do is to improve the throughput of the tilling process by using next generation sequencing methods. So we did a traditional kind of approach using pooled samples. So we looked at 768 samples of genomic DNA that were pooled in a 3-dimensional format. Following this, in collaboration with Professor Sharma and Pratik Gupta at the University of Hyderabad in India, PCR amplicons were produced of 600 base pairs covering 45 genes. We then took those PCR products from all of these pools, normalized the concentration, and then pooled them, and then performed next generation sequencing and SNP analysis. This is what the data looks like. So these are for the very first experiment that we did. Each of these green bars represents the number of times we're sequencing the DNA for the different PCR amplicons. So here you can see these little peaks are the center of PCR amplicons. So you have one PCR product, 2, 3, 4, 5, 6, 7, and so on. This is the coverage, which is quite high, and these blue lines at the bottom represent mutations, putative mutations that are identified by the software. This is a summary table of that, where we're building bioinformatic analysis to find this. And I'll just point out, for example, here at position 2181 in this pseudochromosome, we have a C to T change that is represented in each of the 3 pools, and this is a canonical EMS type mutation. So we found a bunch of these mutations, and we are now validating them. This is another way to look at the mutations. So this is a graphical representation, and the blue arrows show you 2 mutations in gamma glutamyl hydroxylase, which is involved in folate biosynthesis pathway. So all of these little dots represent sequence variations that the computer found, but at a very low level, indicating that they're not real. And the higher frequencies here represent true mutations. So we've now built a pretty efficient pipeline for doing this. Last week, we did a similar experiment with barley. So this is tomato, and we had a collaborator coming from Poland, and we developed an improved strategy for barley as well. In India, they've taken these plants and they've validated that the mutations do exist. So we have a pretty useful tool for analyzing mutations. We've done this now in
this slide, I'm just showing you how we've increased the throughput. So we've done an experiment. That last experiment was, I think, with about 12 PCR products. This is with 45. And we can clearly see the mutations there. And we have here in this example, we've got higher fold pooling. So this is 128-fold pooling. And we see the mutations appear. Our newest experiment goes up to 250-fold pooling, which we think will work pretty well. So we now have a strategy for finding gamma mutations that are large deletions that are caused by mutagenizing seed. Here we have chemically mutagenized tomato seed, and we developed a good method for finding the induced point mutations. We've also been working on vegetatively propagated species, and what we do here is we isolate meristems. So Joanna did an experiment that took several years where she clonally propagated bananas, isolated the shoot apical meristem of these bananas, and then treated them with EMS. After the treatment of this, she then continued to allow the plants to recover for a month and then tried to purify the chimeras by doing multiple rounds of meristematic isolation and longitudinal bisection. So this is one way. Bananas are vegetatively propagated. If you are eating the yellow Cavendish bananas in your country, you know that there's no seed in them. So they have to be propagated vegetatively. These are cartoons of what we think is happening during mutagenesis. So on the top panel, we've got meristematic cells in different colors to show that one model where each cell accumulates different mutations after mutagenesis, and how longitudinal bisection can be used to reduce the complexity of the genotype, the resulting genotypes. Another model which we were testing is called diplontic selection, where the mutations are rapidly fixed to a single genotype due to competition of cell divisions in the meristematic tissues. And it turns out that our data supported model 2 here, that this happens pretty quickly in banana. So we found a very predictable high density of mutations in banana. The density was 1 in 57,000 base pairs. Bananas are triploid, and this is exactly what we would expect based on the data from diploids, tetraploids, and hexaploids. We found rapid dissolution of chimeric sectors. This work was published in 2012. One of the other things that we found was that many of the banana plants, and I think this might be true in things like coffee as well, have accumulated deleterious alleles that are maintained in a heterozygous state. So this is just a short table from our publication showing deleterious alleles that are naturally accumulated. And many of them are in a heterozygous state. And this is a phenomenon called Muller's ratchet, because the plants that are vegetatively propagated cannot outcross, and they cannot remove deleterious alleles from their genotypic background. So we expect, we see this in citrus quite a lot, and we see this in, I think we're going to see this in coffee as well when we look at that. We also see this in cassava quite a bit. Okay, so we think that this is important. In bananas, we saw that all but one of the genes we looked at had these deleterious alleles. And we think that this is very important when we think about things like cassava and coffee and other crops that are vegetatively propagated. Okay, so some things to consider when we think about reverse genetics projects in our lab is that we have many projects are focusing for reverse genetics on chemical mutagens, because they create a large number of point mutations, so they're easy to find, they're very predictable. But forward genetics and breeding tends to use more deletion-based approaches. We have many different technologies that will work for tilling, from simple agarose gels all the way up to whole genome sequencing. If you want to do these kind of approaches, you need to know the genes, and you have to have some hypothesis. So if you're interested in disease resistance, you would have to choose the genes, the R genes that you want to mutate to test for resistance. We do know now with some high precision what to expect from EMS on the plant genomes. We don't know this much for gamma radiation or X-ray. We're trying to address that gap. But nowadays, people have even done exome capture for tilling in wheat. So there's over 10 million mutations that have been discovered and shown in wheat. We can also think of things like CRISPR-Cas9 and other genome editing tools that would allow you to directly mutate in your plant of choice rather than use a random mutagen like EMS. So we can think about different approaches for reverse genetics. Now, I want to get back to thinking about what happens in plants that have this striking phenotype. So one of the things we asked is, well, you know, when we think about mutations, we think primarily that we're changing the DNA. And that's a heritable effect, and we see stable phenotypes. We do know, for example, when we thought about barley a little bit, we know that barley grows in a huge range of latitudes. From the perspective of latitudes, it's the most broadly dispersed major crop. So what we decided to do is not look at the DNA sequence per se, but look at the methylation of DNA in different barley accessions. So here we're not looking at mutations, we're looking at different plants that have been collected from around the world. We're doing this with collaborators at the Gregor Mendel Institute, Magnus Nordberg's group. So we looked at 128 different barley accessions, and we looked at whole genome methylation. And this is just a graph of that, and the arrow here points to a very extreme outlier that grows in Afghanistan. And in fact, it looks a lot like this mutant barley growing in Peru because it can grow above 2,400 meters with good performance. And so what we see here is that maybe plants can use methylation as a way to rapidly adapt to different climatic conditions. And in fact, Magnus's group has shown this in Arabidopsis thaliana growing in Sweden. So we think actually that there may be natural mutations in genes affecting methylation like MET1, CMT3, and we're doing further studies to follow this up. Okay, and then getting on to this, we have to ask ourselves if we're interested in mutations and mutation breeding, which is the field I study, why would we bother to make mutations if there's already good mutations in nature? And what we've been doing is using next generation sequencing approaches to look at natural variation. We've been calling this eco-tilling by sequencing. Eco-tilling is a name for looking at natural diversity, looking at SNP variations in large populations. So this cartoon, this graphic shows you what we did. At the CIAT at Columbia. At CIAT, they have a collection of about 6,000 cassava accessions. So we wanted to develop a method where we could rapidly look at allele diversity in a very large population as cheaply as possible. So we didn't take the 6,000. We decided to start with 1,728 of these accessions and 93 primer pairs, focusing specifically on starch biosynthesis and herbicide resistance. We had a grant and we had 2 students come from Columbia. The DNA was shipped to our laboratory here in Austria. It was lyophilized, and we resuspended that DNA. We quantified the DNA and used free software, ImageJ, to quantify the genomic DNA, normalize the concentrations, and then pool. And we pooled about 250 samples together. So each sample now contained 250 plant genomes. Then we amplified with all of these different primer pairs, and then we pooled the PCR products together and we performed next generation sequencing. So we tried to do this as low a cost as possible, and our goal was to get it down to about 10,000 euros, including the labor costs. And I think we're quite close with that. And we also developed a rapid bioinformatics strategy to ask what the alleles are. This is an example of the data here. So this is on the right panel. This is Lina Kafuri and Daniel Tello. who were the 2 undergraduates who did this work, along with Sneha Datta, who is a postdoc. So she did the next generation sequencing. They did all the PCR, primer design, genomic DNA. They had no experience working in a laboratory before coming to my lab, and they did a fantastic job. Bernard in my group also helped out with this, and this is Pratik Gupta, who did the bioinformatics for us when he was working in my lab. So again, we have this sort of graphic on the top showing you the read coverage, the number of times we sequence a specific base for the different PCR products. And down below, we have a summary of this data. So we found thousands of SNPs. There's a huge amount of data, and we wanted to then find a way to rapidly filter this down. And using a program called SIFT4G, we were able to come up with a handful of mutations, and they're listed here, that we believe are predicted, that we predict are going to be deleterious to the function of genes. And that's what we really care about. Gene mutations, or natural mutations that will affect the function of the genes and alter, for example, herbicide resistance and starch quality. And so this has been quite a big success. We're writing a paper on it now. We worked with Hernan Ceballos' group at CIAT, and there's a Colombian government grant that funded the work. So we're quite happy with that. So we think this is working quite well now in our lab as well. So just to summarize then, some of the techniques that we're using, and then we can have any questions and discussion. You know, the reason we're doing this work and the reason we think that mutations are important is because we have a big problem with food security. In the coming decades, it's estimated that food production needs to double in order to avoid catastrophes. We have a population that's growing rapidly, especially in the least developed countries of the world and in places like Africa. And we also see very severe effects of climate change and variation that have negative impacts on yield. And so we need to continually breed plants in order to maintain and increase yields. So this is an absolute requirement if we're going to keep quality of life and improve the quality of life we have on this planet. When we think about breeding, you need nucleotide variation to breed. There's no way around that. You can create that with mutations. You can create it with transgenics, with CRISPR-Cas9. You can find mutations in nature, natural variation in nature, but you need that. You need allele variation to improve crops. And all of the techniques that I showed you today, just brief examples of, are aimed at capturing and understanding that nucleotide variation, whether it's induced or natural. We know that mutation breeding has been widely successful in both developed and developing countries around the world. I showed you an example from Peru. We've got examples of tomato improvement in Cuba, rice improvement in Vietnam that makes the rice more saline tolerant and has improved the income of farmers by 30% annually. So we know that these techniques work. And we're trying to develop sequencing technologies to offer and provide gains in both the precision and efficiency of the process. We also are doing things in the lab, which I didn't have time to talk about, where we develop low-cost methods, things that you can do yourself. And this is a screen capture of a book we published last year. It's freely available online. You can see in this book, we have low-cost methods to produce genomic DNA from plants. They cost about 1/10 the cost of a kit, and low-cost methods to find SNP mutations in plants. So you can do this for less than 1 cent, 1 euro cent per reaction. So we have some very nice low-cost methods that really complement the high-throughput, high-technology sequencing approaches. And finally, I will just, before I take any questions, I just need to acknowledge that all of the work that I showed you today and explained to you today really involves the collaboration with a lot of people. And our laboratory here in Austria has been, over the years, heavily collaborating with people in different countries who have expertise in different crops to help us develop these methods. And I tried to mention most of the guys we're working with as I gave my explanation. So I'll just leave this slide as is, and I'll just say thank you for your attention.
Thank you, Brad.
Thanks. Yeah. So if you,
Thanks, Brad,
Jeremy?
for your nice presentation. ¿Hay alguna pregunta para él? There are any question for you? We have two or three questions. Tiene que sentarse aquí, no sé si. No funciona. Trate de hablar.
Hello,
hi.
can you hear me?
Yeah, it's good.
Okay, my name is Rafael Orozco. I'm working at the National University. It's a estatal university, public university. Sorry. I have a question. Now we are working with rice. We have our card project. I have a doubt. How many seeds that I need to irradiate to obtain at least one mutant to recover a mutant in M2 generation?
Right. Well, we're analyzing the effects right now. We have data sets on gamma radiation and X-ray radiation in rice. I don't have great numbers with that. From EMS mutagenesis, I can tell you that when you do those experiments, so in gamma radiation, we see about 70% survivability with our dosages, looking at like 150 gray, 200 gray. So this is part of it. When you irradiate or you chemically mutagenize, you kill some plants and you can induce sterility. And with chemical mutagens, it can be quite high. Mm-hmm. So it can be from 40 to 70%. So you have to irradiate or mutagenize more seed than you need. We tend to say that living material, a good estimation is between 3 and 5,000.
3
and 5,000.
Okay.
I'll leave that survive. So this would be if you define like your M2 as the beginning of your line.
Okay.
And you were to do a pedigreed approach, I would want 4,000 or 5,000 M2 seed coming from different M1 plants. Mm-hmm. You know what I'm saying? Like if I was doing a single seed descent, and you could bulk those lines together. Usually, most people bulk a panicle. You know, from the M1. So that would be about it. But I would caution also, and that's why we're developing these tools. When you produce the M2, it would be a good idea to randomly collect some leaf material for DNA. Mm-hmm. And the reason for that is right now we're still developing the tools, but I think in the next 2 or 3 years, we'll have the ability to relatively cheaply look at the number of mutations in rice. We're developing the techniques now. It's still a little expensive, but if you collect some leaf material and you store it, it doesn't cost any money. But you have this data for the future. You could do some tests in the future.
Okay. I have another question. We are making a test for salinity resistance. We are making a preliminary test using Petri dishes to put the seed in the M2 generation under pressure of the salinity test to make us preliminary selection. If some plant is recovered from the petri dish, we'll come to the greenhouse to submit to high conductivity electricity. But I know, do you think it's correct to make a preliminary selection first, or make the selection directly in the greenhouse condition? Because in petri dishes, I have a lot of area I can see to evaluate a lot of plants, a lot of seeds. Right. Because in the greenhouse, it's more difficult to check.
Yeah. What we've done here, and we have protocols for this, we've done a hydroponic screening method. So we have trays, and we grow seedlings up in liquid media where we add salt to do the screening. And it's similar to Petri dishes, but what we tend to see is you have to grow the plants for a number of days before you start seeing browning, and we have salinity tolerant material, and we should have some I don't know if you've got salinity tolerant varieties. So the experiment I would do is take some salinity tolerant material, grow them in your petri dishes, and see how they perform.
Mm-hmm.
If they're obviously greener and you have a good the petri method that you're using works, then I would say it's great. You're throwing away, you're potentially losing material that might be interesting, but you have a very rapid and cheap what we call mass screening technique, and that's very good if it's working good. So if you can recover, the idea is that you can start with 10,000 plants then and find a few hundred that may look like they perform better on the petri plates, and when you move those into the greenhouse, you can do more careful studies, and maybe only a fraction of those are actually really interesting. So you just want to make sure that your original screening pressure isn't too strong in the petri plate. You want to kind of get the right conditions so that you don't kill everything, so you can find some survivability.
Okay, thank you very much.
But you can send, Andres can give you my email, and we have protocols for this as well,
okay.
Different methods that we can share with you. And then I don't know if we have reference material, but if we had some seed, or you could order it from Erie, different salinity tolerant varieties that would serve as controls for your experiments.
We are working in two ways: salinity tests and also a dropped condition. We are using to simulate that drop condition. We are using a polyphenol.
PEG.
Yes.
Polyethylene glycol.
Yes,
yeah.
and also I can use sorbitol, but I have doubt in relation to this. Which is better?
Well, I don't know. We have a colleague here that's done some of the experiments. Again, you can send me an email and we can share our experiences with you, because since I'm more focused on the genomics and less on the phenotyping stuff, I haven't done those experiments myself. But most people tend to prefer PEG rather than sorbitol.
Okay, thank you very much.
Yeah.
Yes?
Pasa el material de posterior a la preselección del plato fértil a las condiciones de invernadero, se ve que seguir utilizando el factor de selección, que sí es una buena idea seguir utilizando.
I have another question. If I make a selection, I select one plant from petri dish, I need to go to the greenhouse condition. I need to maintain the same level of stress in the greenhouse condition.
Well, I would be tempted. The hypothesis is that is a genotypic effect. It's an effect of the mutation. And I would be tempted, what would I do? If I found a plant that had a nice petri phenotype,
Mm-hmm.
I would almost be tempted. I wouldn't want to kill that plant, right?
Mm-hmm.
And it depends how many of those I find, but I would be very careful to make sure that I could produce seed from that plant. Because if I kill that plant, I lose the next generation.
Mm-hmm.
So depending on the speed of it, I would almost be tempted to do it if I had a Petri screen, to take the things that I liked, and let's say 10% or less of the entire population, if I find interesting mutants, put those in the greenhouse, let them recover, produce seed, and then phenotype the next generation.
Okay.
And then I would keep, so then if I have 50 seed, I would test maybe, you could assume that I would assume that it should be homozygous, but maybe it's a heterozygous mutation that's giving you the phenotype. I would self it again and test maybe 20 to 30 individuals from that line and see how they perform under the salinity test. And then I would be tempted to try maybe a few different conditions. Some of the seed at a milder condition and some at a very hard condition, very high saline.
Okay. Thank you very much, Doctor.
Yeah.
Thank you.
¿Alguna otra pregunta?
Hi, I have a question. You were showing, like, when you identify the mutations using whole genome sequencing, that when there are big pieces, you can identify them very quickly, not straightforward. But then, how does it go with the SNPs, not with single mutations?
Right. So when we're doing whole genome sequencing, so we're right now we're trying to evaluate the frequency of large deletions versus SNP mutations. Some studies have suggested that gamma radiation produces mostly large deletions, and we see this. We've seen this in sorghum, in rice, and banana. Now, the advantage of a large deletion like that is that it's easy to see because you have many pieces of DNA that are missing. And so when we do the whole genome sequencing, we only need very low coverage. So each base in the genome, we only need to sequence maybe 4 times. And that means we can sequence more plants in our machine. When we look for SNPs, we tend to want 10 to 20-fold coverage, right? So if we sequence each base in the genome 10 times or 20 times, we have enough data to accurately call SNPs. So in the rice experiment that I showed you, we only could find large deletions because I sequenced 17 plants on a single MySeq run, which is a relatively small sequencing machine. Now what we've done is we've done a high-seq run, so we've done many-fold higher read coverage to detect the SNPs. But in theory, if you do the experiment right, you can detect both SNPs and indels in a single run. It just costs a little more money.
Yeah. But in that case, then, if you think in terms of money, will it be recommended then trying to use these other techniques like such a gamma radiation in order to also ease the detection using these high technology methods?
Well, I think, I think right now, the data we have, it is interesting. If you want to do reverse genetics, I would still say doing a traditional tilling approach with EMS is a really good bet because you're making single point mutations and you'll affect single genes. So you can ask the question, okay, I want to know, does this disease resistance gene, does it have any effect on coffee leaf rust, and I can mutate the gene, test it, and it's a very scientific, hypothesis-driven approach. For these mutation breeding projects that have really started in the 1930s, most people are using mutagens that create bigger deletions, and that may be important because in the field, it's easier to detect stronger phenotypes. And so, it's this very simple approach, and I think what we're going to find, although we don't have enough data, is that many of these are these large deletions. And the nice thing about that is if you're interested in doing phenotyping and breeding your plants and growing them in the field and what we were just discussing, like salinity tolerance, you could gamma radiate very easily, find salinity tolerance, and then later, relatively cheaply, find candidate mutations, but they may be very large. You may have deleted 30 or 40 genes in rice. And so understanding the biology of that is going to be very complicated. Building a tool to help breed that material into other elite rice varieties, that will be very easy. So I think it kind of depends on your objective. If you're more into basic research and you want to understand gene function, probably you want to go with point mutations. If you're more interested in developing stable field phenotypes and breeding new cultivars, you might want to go towards something like gamma or X-ray radiation.
Good, thanks.
Yeah.
Hello, thank you very much for your presentation. My question is, you mentioned something that we eventually could have information about the genes that we are expecting to mutate in order to have a difference, or I don't know if we are looking for salinity tolerance or for disease tolerance. But in this case, how do we select those genes? Because at the end, if we are using gamma cell, we will not know exactly where this is going to happen. What can you advise?
So the idea is that you can make, if you're doing reverse genetics, you want to build a large population so that you randomly have mutations. And so you just then ask the question, is there a mutation in the gene? And the genes are selected usually based on sequence homology. So what we find is that, for example, disease resistance is an easy one because there's canonical, NBS, LRR, leucine-rich repeat genes that are known to be implicated in disease resistance. And if you look at any genome, that sequence, you'll find annotations based on that homology, and you can find several hundred of these genes. I agree with you, though. With gamma radiation, we think that there's a mixture of large deletions and small deletions and small SNP mutations. And so with gamma radiation, you choose your genes and you can basically ask two questions. One, is there a SNP there? But the other question you might ask is, does that DNA exist anymore? In our case with rice, we're missing genes. So when we try to, which is complicated because when we try to do a PCR, what we're looking for is the PCR to fail, right? There's no PCR product. And it's a bit weird because normally if you do a PCR and it doesn't work, you just think, oh, I think the primers are bad, or I don't understand this, I just throw this data away. But you have to think about the data a little bit differently. You need nice controls with the parental material, and you need to be confident in your PCR, but you could ask the question, in my mutants, if I study 10 or 20 genes, do I ever find the case where the PCR doesn't work for this gene? Maybe it's missing, maybe it's been deleted.
Okay. And another question, you mentioned with the example on banana, that when you mutate the meristem, you select the cells that are mutated. How do you do this? Using a selective method on the tissue culture or something like that?
No, actually, we're letting nature do that selection for us.
Okay.
But what we're doing is, the problem that we have with banana, or if you're doing any kind of multicellular tissue culture, is that you're basically working with a set of cells, right? Unless you do like a single cell culture, right? Which is possible in cassava, it's possible in coffee. I think that's probably the best way to try. In banana, we didn't have that system in our lab. So we have a meristem that has many cells, and that meristem, each cell we thought was getting different mutations. What we learned with EMS was, in bananas, very quickly, every progenitor from that line shared exactly the same mutation. And what that meant what we think that means is that when we mutated the meristem, it's triploid, that most cells, right, most cells contain mutations, but there wasn't a big difference in that the cells could survive, and that some cells were slower at dividing because of the mutations. And so there's a competition for cell division, and after 1 month, we had basically a central zone of the meristem that was homogeneous because one cell was stronger and outcompeted the other cells. And we think that's what's happening. But to get around that, what you do is you would cut the meristem in half, and every time you cut the meristem in half, you have 50% less cells. So the complexity reduces by 50%, and you do that 4 or 5 times, and what you see is that the cells become homogeneous. But there was no thoughtful selection on that. We just let the cells do it. And I think because they're triploids, it worked. And when we tried to do this with diploid cassava, which already had natural mutations in it, I think what we ended up doing is killing cells. So we were only selecting cells that accumulated 0 mutations. I think we caused a problem for ourselves with using diploid material in that case.
Okay, I am asking this to you because we are using on tissue culture on rice using Kalai in order to try to select some cells, but the point here is not to kill the cells that have the mutation eventually, using the tissue culture and the selection.
Yeah. Oh, please continue.
No, it's okay.
I was just going to say, in that case, I mean, the expectation is when we mutate, like I think with rice, the mutation should be heterozygous if you mutate the calli. And rice, is pretty isogenic, it's self-fertile, so it should be pretty homozygous. And in that case, what I expect is that you'd mutate one of the two copies of the gene, and that all the different cells would accumulate mutations, and the fitness of those cells would vary, but not dramatically. And so when you recover material from that, and you grow and you regenerate a plant, every plant should have mutations.
But in order to eventually analyze genetically or using these molecular markers or whatever, those genes or those plants, is better to analyze them on M3 or M2 plants rather than in plants regenerated from the calli, right?
Yeah. Well, it depends on how many cells went into making that plant.
Okay.
Yeah, so if you could regenerate a plant from a single cell event, then you could immediately screen, genotypically, you could immediately screen that first generation.
Okay.
You could just take leaf material growing. But I don't know, and I haven't done those experiments myself, but I've talked to a lot of people about that, and it's unclear to me. In some cases, I think you have to be very careful that you're not producing a plant from multiple cells.
Okay.
In which case you would want to let that plant regenerate and produce seeds, and then begin your phenotypic and genotypic screening in the next generation.
In the next generation, yes, it's better. Otherwise, it could be a waste of time and loss of money. It's quite expensive.
Yeah, I think it's difficult. I think it's interesting to try that, but you're going to spend some money to learn whether or not they're chimeric or not. And I was talking with Andres when he was here about maybe doing this with coffee leaves in the M1, because you have to wait so long for coffee.
Yes, it's
but,
true.
You would be spending thousands of euros just to ask a question whether or not it would work. And it's a difficult experiment to justify.
No, thank you very much. This is all I have to ask.
Okay, thank
you. Bye.
So, Brad, thank you very much. It was a nice
thank
presentation and very informative. So we want to keep in touch with our experiments and your laboratory in Vienna. I know that you are going to leave this year, but maybe someone else is going to continue with your job.
Yeah, well, I hope so. And we're trying to build, and we're doing a lot of interesting experiments in the next few months before I leave. And my goal is that we can set this up and that the lab can continue to really help support these kind of activities that you guys are involved in, and using gamma radiation and different kinds of mutations to do functional genomics and crop improvement. So I do hope, and me personally, I'm happy to provide any advice I can, whether it's this year or next year, wherever I'm working next. So I hope you stay in contact with me as well as the lab.
Yes, we will keep in contact, and we can also extend an invitation to visit us here in Costa Rica, to our university.
It sounds great. Yeah, I would look forward to that opportunity and to discuss things further with you guys.
Okay, thank you very much.
Great. Thanks, Andres.
Good afternoon. I know that
Okay.
there is almost 6 o'clock.
Yeah, I don't know what. Oh, it's 4:30 there now. It's not bad.
So you can go and work a little bit more.
I still have some work to do. Yes.
Okay.
All right. Great.
Have a nice day. Thank you. Bye-bye.
Bye-bye. Bye.
De nuevo, muchas gracias a todos por su presencia. Espero que les haya sido
Muy buena.
la conferencia.
Sí, claro.
Como les digo, nosotros acá en el grupo tenemos un proyecto de inducción de mutaciones en arroz y otro en café. La parte más interesante para mí es la detección de esas mutaciones y no tanto la inducción. Eso es un poco más técnico. Por eso que traté de hacer esta conexión con el grupo de investigación en Austria. Y para los que estamos involucrados en esto, sería esta una oportunidad para conformar un grupo trabajo, un consorcio tal vez de inducción de mutaciones en las 3 universidades y avanzar en el siguiente paso, que es la detección de esas mutaciones. Yo visité los laboratorios en Viena, el equipo que tienen ellos no es nada sofisticado y diferente a lo que tenemos nosotros aquí. Máquinas de PCR, tiempo real, secuenciación, y lo podemos hacer. Y el costo no es tan elevado, son como $10,000, 10,000 euros para analizar unos mutantes. Menciona estos 2 libros, un libro de protocolos para la detección de selección de salinidad y otro para la detección de mutantes a bajo costo. Los 2 están disponibles, están en acceso gratis. La universidad tiene acceso a la base de datos de Springer y si alguno no pudiera, pues me contacta a mí y yo los puedo enviar. De nuevo, muchas gracias a todos.
Gracias.
Vamos a ver lo del curso.
Estamos manteniendo el movimiento de selección. De la casa de todas maneras, es una M1. Esa presentación de minutos. Voy a hacer el mantenimiento. Una parte de la selección y de la clase de otra parte de la casa. Sí, para eso hay que hacerlo.
Bueno, está bien, por aquello.
Bueno, gracias
Bueno, corrió ¿se va a estar en la...
Sí, ahora no está.
bueno,
Después de aquí me voy y voy para allá.
cualquier cosa está fueron en el software ahí, todavía está, ¿verdad? Sí, bueno, probamos primero nada más los volúmenes. Primero me dijo que mejores cosas, porque tal vez la concentración de primer no está importante tocarle. Y la temperatura de agregación, como yo ya había hecho aquí las pruebas, la primera prueba fue de volumen, 10 y 20 microlitros. Y hubo algunos resultados que estuvieron interesantes porque las TM sí están muy similares, pero en el equipo sí detectó como variantes.
Dentro de su material.
Sí, entonces, ahora pensando, yo no tengo esa duda. No sé, digamos, es que son TMs muy bajos.
Lo que podemos hacer es, se puede correr tal vez en este secuenciador capilar, para ver si se ven esas diferencias de esas dos variantes.
Sí, de hecho yo traje los productos, me dijo que lo pudieran en la garosa para ver si, porque, bueno, lo que hicimos es que las muestras en 20 microlitros sí estaban con mucha mayor concentración de alcohol, entonces que lo pudieran en la garosa, igual que la reacción, ¿verdad? Entonces, ahora hablamos para
sí, porque ahora el siguiente paso, yo le voy a escribir a él, copia usted. Hasta luego, yo Marta, muchas gracias. Para que nos dé recomendaciones de cómo diseñar los primers para HRR, ya usted tiene las especificaciones, pero entonces lo que le habían pedido, que identifiquemos una pieza 15 para diseñar los primers que tienen que ser como 1500 y después ver diseñar los de HRR. Yo lo voy a escribir a él. Y me dijo que si hay posibilidad de que usted vaya. Lo que tenemos que hacer es conseguirle el tiquete de los planes. Chao. Pues como le dije, más o menos como 1.000 euros por semana, por mes, por lo menos para aprender la técnica, podría ser un mes que vayamos pensando en eso y veamos si hay alguna posibilidad de financiamiento de aplicar. Yo le mandé las opciones de la beca del Senado. Ahora hablamos. Señor Alejandro, gracias. Sí, yo lo busco. Chao.
Señores.